Hello, my name is Melanie, and I will be talking about our current work on decoding the adaptive immune system using deep generative models. T cells are one of the key players in our adaptive immune system, identifying and fighting diseases. When we get exposed to a viral infection, antigen-presenting cells in our body consume the proteins at the surface of the virus called antigens, break these down, and present these on the cell surface. This is where T cells come into play. Each T cell has distinct T cell receptors, or TCR, that will bind very specifically to a presented antigen, triggering clonal expansion. We can measure expansion of a given TCR by sequencing a blood sample and counting how many times we read the unique DNA sequence of each TCR. That is what we call a TCR repertoire, a vector of TCR counts similar to a bag of words representation of a document in topic models. But which TCR correspond to which disease? In practice, we don't know that mapping. And several factors beside disease exposures influence the counts, including demographics or undesired artifacts such as sequencing depth. So in this paper, we present AIRIVA, a deep generative model, and more precisely, a domain invariant VAE, variational autoencoder, to decompose the TCR immune response signal. Training data consists of TCR counts and repertoire labels, which may include diseases, demographics, or sequencing depth. TCRs are then mapped to independent latent factors through a factorized latent representation, which are subsequently used for prediction. Finally, by intervening on the disease targets or sequencing depth, similar to toggles of a simulator, we can generate realistic in silico repertoires. In our paper, we present two infectious disease case studies for COVID-19 and herpes simplex virus, or HSV. In both scenarios, AIRIVA learns a factorized latent representation of TCR repertoires where each axis corresponds to a human interpretable concept. AIRIVA is able to make robust disease prediction across different subpopulations, in contrast to state-of-the-art baselines. We show that AIRIVA can also be used for generation of realistic in silico TCR repertoires, either conditionally or via counterfactuals. Finally, we empirically demonstrate that AIRIVA is able to recover true TCR antigen associations, which we validate using external TCR annotations from the wet lab. If you're interested in TCR repertoires or deep generative models, please come to our poster or reach out. Thank you all for listening, and thank you to my amazing collaborators within Adaptive Biotechnologies and Microsoft Research.